disparities and global health. He currently leads international cancer consortia that study first cancer in BRCA1 and BRCA2 mutation carriers, and prostate cancer in men of African desert in North America, the Caribbean, and Africa. It is an honor also to share the chair with me, Dr. Ahmeda. Thanks so much. Please.
Talk to you today about cancer genomics and what we can learn from Africa. As you know, cancer is an inherently genetic disease, and it's hard to imagine that we can really understand the etiology of cancer or make any headway in the prevention and treatment of cancer without having underlying knowledge of genetics. So this is a fundamental principle that I think most of us, and most in the genetics and cancer community, would agree. But the problem, of course, in Africa is that we have many problems. And I'm talking primarily about sub-Saharan Africa, where we have very limited cancer therapies, we have limited access to drugs, we even have limited availability to morphine among people who need palliative care. So it's hard sometimes to make the argument that we also need cancer genetics and genomics in Africa. But I'd like to make some of the points about why understanding cancer genetics and genomics is important and how we can learn from it. So let me just make the point first that cancer is a huge burden to the continent of Africa. In the next 2 decades, cancer deaths will nearly double. By 2035, about 1.2 million Africans will die of cancer a year. And currently, more individuals in Africa die of cancer than of malaria. So we're talking about a very big public health problem. So what can we do to understand cancer genetics and genomics in Africa? Well, and why do we want to do this? So first of all, we can learn from Africa. Africans have a unique variation in risk and phenotype and unique disease etiology and natural history, so we can be able to learn from those observations. And also, we have the ability to understand by studying Africans what disease might look like across the African diaspora, and particularly in African Americans, where cancer risk and outcomes are unique and usually unfavorable. It's also important to develop genomics in Africa because we know that developing a new implementation of new technologies has a lot of benefits. The creation of knowledge is certainly one of those, but the development of new technologies in a place like Africa can be important social and economic drivers and disruptors. So just think of the cell phone and how it's changed the structure of communication. We could think of the same kind of thing in a genomics setting. It can foster innovation and investment in high tech. It can stem the brain drain that we often see because there are bright people in Africa who need to stay but don't have such much opportunity. And we can address unique biological and clinical problems. And all of these things are becoming more and more feasible because of cost and technology availability are improving. So just to put a point on why I think it's important and what we can learn from Africa and how we might be able to improve the African situation, I wanted to give you 2 examples. One in prostate cancer, which is the highest heritability of any major cancer that we know of, and also is the leading cancer in men across sub-Saharan Africa, as well as in North American African Americans. And also to give you an example of BRCA1 and 2 associated breast and ovarian cancer, which is a inherently genetic disease, but it is clinically significant, but also preventable. So let me start by introducing you to our study, the MADCAP study. This is the Men of African Descent and Carcinoma of the Prostate Network. This MADCAP network incorporates 17 centers in sub-Saharan Africa and North America, 11 of which are funded by the NIH through a UO1 consortium. The coordinating center is at Harvard. We have a tissue biobank. But all of our genomic analysis and biosampling are done in Africa. None of the samples and tissues leave Africa because we're concerned about developing capacity for doing this kind of work in Africa. So all of the biosample, biobanking, and genomics works are done on the continent. So here's my first example. This is a population genetic and GWAS data example. to show you what we might be able to learn from Africa in the context of genomics. So this was a study of prostate cancer and trying to understand why African Americans have such high rates of prostate cancer and such poor outcomes. And as you may know, there have been many GWAS for prostate cancer. In fact, it probably has the most GWAS hits of any major cancer, probably 130 to 180, depending on how you count them, GWAS hits. Almost none of those have been replicated in African American populations that we've tried, but very few of the GWAS hits replicate. That's not an unusual situation between European, Asian, and then African populations. But we thought we'd try to understand why we might be able to be not replicating, and also to understand what kind of new hits might be out there. So we took 26 prostate cancer GWAS hits and got 68 independent SNPs in these regions where the hits were found in European or Asian populations. We also incorporated with those 1,000 Genomes Project and sequence data from 38 African populations from Sarah Tishkoff's lab using the Illumina 1M Duo. And so we did a few analyses to understand what's going on at these prostate cancer potential loci. We evaluated a number of different parameters, not going into detail here. FST, we computed a genetic risk score, we computed a genetic disparity contribution score, and we also used the composite of multiple signals, CMS statistics. So the few statistics here that we used employed to evaluate the data. So here are the first results. So we have 68 loci in 64 populations. On the y-axis are the SNPs, and the x-axis is the population. And what you can see is different frequencies according to population and by SNP. And I thought to make this presentation easier, I would just walk you through one example of a SNP that came out as interesting. So I've highlighted here RS7584330, which is a SNP that was identified in a prostate cancer GWAS, which has no known function. It's not clear what it does, but it's very consistently found in prostate cancer GWAS in European populations at least. And what you can see with RS7584330 is that it is a SNP that is associated with increased risk in African populations, and it's also found at a high frequency in populations with greater prostate cancer risk. So that already is kind of interesting. We then computed FSTs, genetic distances, and we find down in the left-hand side the distribution of these FSTs suggests that there is some evidence for a high FST between European and non-African populations. So there's some larger allelic differences that this SNP has, particularly between Africa and Europe, but not so much between West and East Africa. So already we're learning that this SNP has got something going on in terms of European versus African populations. We also did two computations. We did a genetic disparity contribution on the left in panel A, and you can see again our SNP 7584330 is up toward the top. It shows that this SNP provides greater risk in Africa. That the risk is conferred among Africans, although there are some SNPs that confer even greater risk in Africans and other SNPs down below in the gray that should confer greater risk in Europeans. So there is a distribution of how the SNPs look in terms of their contribution to risk of prostate cancer, but 7584330 seems to be a prostate cancer risk SNP primarily or more in African populations than in European populations. On the right-hand side in panel B, we also did a computation of all the SNPs that we had and asked, genetically speaking, of the risk SNPs that we studied, who has the greatest risk? What populations seem to have higher risk of prostate cancer? And what you can see is that the SNPs at the top the populations at the top of this list in black tend to be from West Africa. So our calculations also tell us that West Africans, at least genomically, seem to have higher risk of prostate cancer than East Africans or non-Africans. And that's actually fairly consistent with what we've seen in the literature. This is data from a survey sampling screening study in Accra, Ghana. And what was found is that the Ghanaians have the highest prevalence of prostate cancer of any population that's been studied in this way to date. Certainly much higher than US Caucasians, but even higher or as high as African Americans in the US. So our genomic analysis tell us that prostate cancer is a highly genomic disease affecting West Africans significantly, and in fact, the epidemiological data supports that West Africans may have extremely high prostate cancer risk, perhaps because of genomic underlying susceptibilities. We also did scans for positive selection in this same data set, and you can see that our SNP 7584330 is up high in terms of being under selection in Europe, and not as high in terms of selection in West Africa. So this is a SNP that has its effects in African populations in terms of prostate cancer risk, but the selection that we seem to observe occurred in Europeans. So why is that, and what does that mean? So here's 7584330, and the CMS scores in Europeans on the top, Japanese and Chinese in the middle, and Yoruba in the bottom. And what you can see is that the CMS score suggests that the selection really occurred in Caucasian populations. So the summary we find after looking at all these data is that the risk allele frequency for the 7584330, the one that confers risk of prostate cancer, is higher in Africans versus non-Africans, but it's under positive selection in Europeans. And so the question is why? And so what we found is that this SNP is adjacent to a gene called melanophilin, which is involved in pigmentation dilution. It's also right near the prolactin-releasing hormone, which affects male fertility. So we can infer, perhaps, that the haplotype protecting against prostate cancer may have hitchhiked to high frequency in Europeans. That is, there's an allele that is not associated with prostate cancer. It happened to fall on a chromosome that is associated with pigmentation dilution, which was selected for advantageously in Europeans as Europeans migrated out of Africa. And so we're beginning to pick up a story here that suggests that African populations have, when taken through the Middle Passage and during the slave trade, tended to carry a risk allele that is associated with prostate cancer, but was not selected against because they never underwent the pigmentation dilution selection that Europeans did. So again, this is a story. It's not been confirmed by basic biology or anything like that, but what it helps us to see is that there might be data that we can use about population genomics, comparing populations across geography, and particularly identifying interesting, potentially causative, potentially associated GWAS hits that might explain why some populations like African Americans or Africans may have very high rates of prostate cancer, and explain some of the very high heritability that we observe for prostate cancer in the African population in particular. So it's just one story, but it does teach us that we can learn something from Africa. We can learn something by comparing these populations that can be biologically and genomically quite interesting. So let me go to the second example. The second example is BRCA1 and BRCA2. As you know, these genes were identified over 20 years ago through linkage analysis, through family studies, and were found to confer extremely high risks of breast and ovarian cancer. And now we know that they also confer a very high risk of prostate cancer and others. This slide just shows you that the estimates of risk are quite high. So a woman who's inherited a deleterious BRCA1 mutation has about a 60% lifetime risk of developing breast cancer and a 40% lifetime risk of developing ovarian cancer. Those risks are slightly lower for BRCA2, where the risk of breast cancer is about 50% or 20% for ovarian cancer. So these risks are not 100%, they're not complete, but they're extremely high. And what that means is that these mutations should be actionable. We should be able to do something about this risk because it is so high. The other thing to notice is that there's a lot of variability. So this plot shows you on the vertical bars the confidence intervals around individual estimates from different studies around this meta-analysis, which came up to the final risk estimates that were done. But these vertical bars suggest a huge amount of variability in risk. Some individuals may actually be at fairly low risk, and some individuals may be at extremely high risk of breast or ovarian cancer, even though they inherit the deleterious mutation. And in fact, what we observe is even within the same family that has the same mutation, we see great variability in terms of risk of breast or ovarian cancer. So the question is, what does that do to? How can we begin to understand that? And is this variability also associated with ethnic or geographic ancestry? So the first thing to note is that most of the studies that have been done have really been done in whites. And this is not a surprise to anyone because this is the pattern throughout genomic analysis. And the SIMBA consortium is a consortium that has about 45,000 BRCA1 and 2 mutation carriers that we work in. And as you see, of those that are Asian, Hispanic, Latino, or of African descent are only about 6,000 of the 44,000 mutation carriers that we have. So it's really a big problem in trying to understand what kind of effect these mutations have in non-European descent populations. So we tried to address this by developing a thing called the BRIDGE study. BRIDGE stands for the BRCA1/2 International Diversity by Geography and Ethnicity Consortium.